Okay, let's take a look at a comparison between the template strand of DNA, the coding strand of DNA, and the mRNA that is produced from that DNA through transcription. In the slide, you can see the template strand right in the middle. Here's the 5' end, and you can see the sequence of bases right here over to the 3' end. Now remember, the coding strand, the second strand of DNA, is going to be anti-parallel, so the 5', 3' are opposite, and it's going to be complementary. So A's with T's, T's with A's, C to G, and G to C. Now, when it comes time to transcribe this DNA, to transcribe a gene, that gene is located in the template strand. So during transcription, an mRNA will be made that is complementary to the template strand, with the exception of the uracil-thymine swap. So if there's an A in the DNA, there'll be a U in the messenger RNA. If there's a T in the DNA, there'll be an A in the messenger RNA. C goes with G, and G goes with C. So the cell creates the messenger RNA from the template strand. And now, if we compare the messenger RNA with the coding strand, we can see that they're a match. Not a complementary match, but an actual match, with the exception, again, of the uracil-thymine swap. So, thymine uracil, A, G, G, C, C, and so on, all the way down. So, the template strand is the strand that has the gene in it. But the coding strand is actually going to show us a sequence that is a match to the messenger RNA. Now, the RNA polymerase enzyme actually exists as two separate units in the cell that come together and become active during transcription. Remember that we are using bacteria as our model organism here, so I'm going to point out where there are differences in eukaryotes a little bit later. The bacterial RNA polymerase has what's called a sigma subunit and a core. The sigma part of the enzyme is the regulatory subunit, and the core is the catalytic subunit. So, while they are separated from each other, the sigma subunit is responsible for recognizing, in the regulatory region of the gene, a region called the promoter. Sigma is going to come in and bind to this part of the gene, this part of the regulatory region called the promoter. It will come in and it will bind to the promoter, and then it will guide the core into place. Remember, it's the core that's catalytic. The core actually contains the active site. And the active site, as we'll discuss, actually has several different regions in it, several different channels in it that are going to be involved during this process of transcription. Transcription can be broken down into 3 phases, what we call the initiation phase, the elongation phase, and the termination phase. And we'll talk about each one of these separately. On this slide, we can see a representation of the RNA polymerase enzyme in bacteria. Now you can see this term holoenzyme over on the right-hand side. This is a depiction of the fact that both the sigma subunit and the core subunit are depicted. You can see the DNA in red, and you really get a sense of just how large RNA polymerase is by comparing it to that DNA molecule. Again, the sigma subunit is shown in blue, and the core is shown in green. Like all enzymes, RNA polymerase is big. It's a big protein. Sigma, the regulatory portion, recognizes a sequence of bases in the regulatory region of the gene called the promoter. The core is then guided into place by sigma so that it is in the right location to begin transcribing the gene. So when sigma binds to the promoter, it's actually binding to two particular regions of the promoter. And we give these names. We talk about the -10 box and the -35 box of the promoter. Now, sigma is actually binding to the coding strand. And then it's going to guide the core in so that it's at the right location on the template strand to begin transcription. The reason we give these promoter regions these funny names, -10 box, -35 box, is because of some directional language that we use when we're talking about a gene. We talk about the +1 site in a gene. The +1 site is the location of the first base that is going to be transcribed. So it is the first base of the coding, the protein coding region of the gene. That's +1. The minus designation in the promoter tells us that instead of moving down the gene, or what we would describe as downstream, we're located upstream. We're into the negative numbers. So if we count back from the +1 site, if we count back to the -10 site, so from +1 we would go to -1, -2, -3, all the way to -10. That's the location, the first location where sigma is going to bind. And then if we keep going, -11, -12, -13 bases, -14 bases, we'll hit that -35 site. That's the other place where sigma will bind. So sigma comes in and hits two landing spots, if you will, on the promoter region of the gene. So this upstream, downstream language is important when we talk about transcription. Upstream meaning the DNA that is, if you will, behind the enzyme. The enzyme is going to move in the other direction. It's going to move down the region of the gene that codes for protein. So what's behind the enzyme is upstream from there. Downstream refers to the direction the enzyme's traveling in. That's what's ahead of the enzyme, what is waiting to be transcribed. When sigma comes in and binds to the promoter at the -10 and the -35 box, once it's bound, it's there to help guide the core into place so that the core is sitting on the template strand in just the right position to transcribe that very first base at the +1 site. Now this is another drawing that depicts the RNA polymerase enzyme interacting with a DNA molecule. It looks a little bit different. If we start at the top, the DNA is still in red, but they've drawn it with a little more detail. Same with the enzyme. The sigma portion is still in blue, the core is still in green, but they've actually depicted for us all these little circles that represent the individual amino acids in this large protein. The DNA has two strands in it. They've depicted those as red and pink. So sigma is going to come in and bind to part of the regulatory region of this gene, the -10 and the -35 positions upstream from the +1 site. And it's going to guide this core into place so that as the core starts to move downstream, the very first base that will be transcribed is the +1 base. Now, in the middle of the drawing, they've drawn for us the promoter region of this gene. Remember, sigma is actually going to attach to the coding strand. And they're showing us both the -35 sequence and the -10 sequence, where those binding interactions will occur. Now look at the bottom of the drawing. What they've done down here is they've cut the core in half so that we can peek into the inside of this three-dimensional protein. The darker regions that they've drawn here, those represent actual hollow places inside the core. So we've sliced it in half so we can peek into these tunnels inside the core. And these tunnels become important because as this enzyme moves down the coding region of the gene, so here's the +1 site, it's going to start moving this way and it's going to do its transcription work. What we're going to see is different things coming into and exiting out of these tunnels. So let's start with the initiation phase of transcription. Sigma is going to come in and bind to the promoter region. The core is going to be guided into place. The double helix of DNA will be opened to separate the template and the coding strands. The template strand is going to be threaded into one of the channels inside the core. The activated nucleotides, the NTPs, are going to enter through a different channel in the core, and the sigma will now be released. We don't need sigma anymore. Sigma is the regulatory part of this enzyme. It's the core that's going to do the catalytic part. So sigma got the core where it needs to go. It got it all set up. Now sigma can go. Sigma can let go and leave the process, and the core can begin to do its work. What they're showing us in this drawing, again, is the entirety of this enzyme. So they're showing us the sigma and the core, and here's the DNA molecule. And just as they did in the last drawing, the red portion of the DNA and the blue portion of the DNA distinguish what's regulatory and what's not. So this blue area is the promoter, the regulatory portion of the gene. And then the red represents the downstream DNA over here, where the protein code is. So the +1 site would be right at that junction. Sigma binds. It brings the core into place. We need to separate the two strands of DNA, and you can see that happening here. The template strand has to be threaded in to this core part of the enzyme. It has to be threaded into one of these channels. And that's what they're showing us here. The NTPs are going to be coming in through another channel. Remember, these are the building blocks of RNA. So the enzyme can be adding these in one at a time and building the yellow RNA that you see here.